So, good morning, everyone. So, right now we are going to start our tutorial 3, and today you will be having a quiz from 9 to 9:15. So, we'll complete this tutorial by 9. And again, for Q&A, you can join us back after your quiz at 9:15 to 9:30. So, we'll start with this part. So, I'll be covering the initial parts of the slides, and Soumidhi Mandal will be covering the other part of the slides. So, can you hear me all? Soumidhi?
Yes,
Okay. So, what we'll see in this slide. So for cloning the DNA, like to make the several copies of the DNA, so we'll show you the steps that are common in all the following procedures. So whatever you do, if you need the more copies of DNA, so this is the general principle that we usually follow. So what we basically do, from the source DNA, we take the gene, and so we enzymatically fragment that gene, and then we put that gene in a cloning vector. So cloning vector is basically a small piece of DNA that we usually take from a higher organism, virus or a bacterium. And so once we insert this gene in this cloning vector, so the resulting product that we get is the DNA construct. So then we put this DNA construct inside the host cell so that it can get the microenvironment, so that and further it can synthesize its own proteins. So because as we told you in the initial tutorials that you must be remember of the central dogma that from DNA we get the mRNA, and from mRNA we get the proteins. So this is the same thing that is applying here also. So once we insert the gene, the end product of the gene will be the protein. So once it's inside the host cell, so we'll get the proteins. So in this slide, so this is the basic general procedure that we usually follow in all these cloning experiments. But how do we confirm that but we should be confirmed that the gene and that we have inserted in the cloning vector is indeed that part that we usually want. So what we basically do, after we make a DNA construct, then we fragment that construct. That is a restriction with the help of restriction enzymes. So we digest that construct. So for example, we are using EcoR1. So this is the PET28A. This is the name of a vector. So you can see in this electrophorus gel on the very left hand side, you can see the DNA ladder. So which basically show you the base pairs of the DNA. So just to quantify, because when we use this vector, so we prior hand we know the base pairs. So we just want to confirm that the DNA construct that we have made, it's indeed that or not. So what we do, we basically digest that vector, and thus we just ran the electrophoresis, and you can see the bands that on that. So we have one ladder 1, 2, 3, and 3, 4, and 5. So in the ladder 1 we have the DNA ladder. So just to compare the base pairs. And on the land 2 we have the digested part. So in digested part you can see that there are fragments, and on the left hand side you can compare in which base pair they are lying. So we know these base pairs prior hand before starting these experiments, and you can see on the undigested part, like on the lane 3, that it is undigested. You can see the lighter band and the thicker band. So this is the original part, undigested part, and after digestion we get this part, lane 2. And so because lane 4 and lane 5, it shows the second part, because it may be possible that there may be multiple fragments of that vector also. So we can confirm this with the help of this agarose gel. So if we want to make a large amount of DNA, so what we basically do, we use the PCR. So you must be read about this thing in the previous tutorials, and in this class also. So he has taught these things earlier also. So in order to make a large quantity of DNA, so we use the PCR. So basically, so if the DNA is very small and you want to do some kind of experiment, you need a significant amount of DNA. For that, you need to amplify that DNA so that you can get a significant amount of DNA. And these are the basic steps of the DNA. So these are the ingredients, like we have the DNA, we need the sample, we need the primers, we need nucleotides, we need the Taq polymerase, which is the enzyme which synthesizes the DNA, and then we need the buffers, and this is the thermocycler on the bottom machine, and this on the bottom, and you can see the PCR machine. This is known as a thermocycler, and on the very right hand side, you can see the cycles of PCR. So we have the three phases of PCR. We have the denaturation, annealing, and the elongation part. So this is basically a basic procedure of this PCR. So in order to, so we also need, so this, we just also want to know that what is the amount of DNA that is amplified. So that part is known as the quantitative PCR. You can see the real time PCR. So what we need in that part, as we know, we need PCR can amplify the DNA. So if we have the mRNA with us, then we have to convert this mRNA into DNA. For that we need the reverse transcriptase. Then we need the DNA polymerase, and we need the buffer, the DNTPs, and from which we can make this CDNAs, and we need the primers also, because this part is totally unknown to us prior hand that from mRNA which primer will work or not. So if we have the gene of interest with us, so what we can do is that we can put the sequence specific primers that we know prior hand that if we want this thing, then we need to add these kind of primers. But if we are totally unknown, then we can add the random primers also, so from which we can get the CDS. So this is known as the basically quantity PCR. So in order to quantify the amount of DNA, how much DNA is quantified. So basically, how does it work basically? So in order to quantify, what we do basically, so along with the DNA, we have the probe also. We have probe. We have in the probe we have the fluorescent dye, and we have the quencher. So quencher, what it basically do, as long as this fluorescent dye is with the quencher, so it will not emit any color. So as long as the Taq polymerase, it will start making the DNA. So what it will do, it will release the fluorescent from the probe. So it will release the separate the quencher from the fluorescent part. So once the fluorescent will be separated from the quencher, so it will emit the fluorescent light, and we can quantify that dye. So from that we can get an idea of that how much DNA is getting quantified. So this is basically a general principle that we have already seen in the previous slides, that first of all, for the making of CDNA, so if we have the mRNA, then we need to put the reverse transcriptase and the other components like buffers, and then we need to do the amplification of that mRNAs, and then what we do then, last we ran the gel lactophoresis just to quantify and just to verify that what we need is actually that part or not. And this is the actually somewhat detailed part of this complementary DNA synthesis, how we make this DNA cDNA from the mRNA. So you can, on the right hand side, you can see that we have the mRNA on the 3' we have the poly A tail, and the opposite to the poly tail we have the primers, which are the poly DT, because opposite to it A we have the T, and then this primer synthesizes the sequences corresponding to the mRNA. So once the sequence is built, then we need to digest this mRNA. We need to remove this mRNA. So for that we need the RNase enzyme that because once this is basically a junction in which we get this mRNA as well as the DNA. So we just need to separate this DNA from the mRNA. For that we need an enzyme. So those enzymes are basically RNAs which can separate the mRNA from the DNA. So once we separate this mRNA, then we use the polymerase enzyme so that we can get the complementary strand of that template strand. So that's why it's known as the complementary DNA synthesis. So then for using these DNAs for our experiment, so we also need to know what is the melting temperature of these DNA. So at what temperature we can separate those DNA. So for that, this is the basic formula to calculate the melting point of the DNA. So you can calculate in this slide we have given one example. So can you please tell me what is that melting point of this sequence? Anybody? Anybody, please calculate this the melting temperature of that this sequence. Formula is given on the top. You just need to count G, C, and T, T and A and T, and you just have to put those numerical values in this formula, and you just have to calculate the melting temperature. Anybody? Can you speak a louder?
56.
56.
56.
58. Okay, so you can try these kind of examples. So in this part, Soumyadeep Manda will take over from this part. Soumyadeep, can you please take over from this part?
You can keep sharing the screen.
Yeah, sure.
Yeah, so as you people can see, can you show the next image? So we have a vector here, circular DNA, and it has been digested with EcoR1 and HAE3, which are two restriction enzymes, and the plasmid of 100 kb. So as you can see in the figure, double digestion will give four bands. Arrange the plasmid sites on the plasmid. So as you are seeing that the this is the entire circular DNA that you have. The two E's represent the restriction sites for the EcoR1 restriction enzyme, and the 2H they give for the HAE1. So once you have set them up accordingly for the size given from the from the agarose gel band, which are the four bands for EcoR1 HAE1 are 40, 30, 20, and 10. If you arrange them accordingly, you shall be getting a following pattern, and the sites for the particular restriction enzymes will be as follows: EcoR1 70 plus 30, HAE1 60 plus 40, and the double digestion will be 40 plus 30 plus 20 plus 10. That is how you design a vector and do the vector mapping. Next slide, please. This is also a same question with the same idea that you are restricting a particular gene, and then upon partial digestion, what will be the length in kb of all possible DNA fragments obtained. So almost using the similar kind of idea. Anybody, can you tell me the answer?
2 kb, 3 kb.
Yeah, go again. How many possibilities can you have? Just like how many possibilities will you have?
How many possibilities in the sense? The answer should be D, lane D.
No, the answer is not lane D. See, there is upon partial digestion. Every single line that you see, the dotted line on the straight linear line given, it is a site for restriction. So if you are digesting your particular DNA, if you're digesting a particular DNA and give it a thought that all the sites are not being restricted. Okay, now if you could restrict all the sites, there are one, two, three sites and possibility of one, two, three, four fragments, right? But maybe all the sites are not being digested at the same time. Maybe there is just one single digestion. So how many fragments do you get? You get two fragments. There are two digestion. You get three fragments, and with three digestions you get four fragments. So you have got like 4 plus 3 plus 2 plus 1. That is how it is going, because you have to consider the possibility that not every single restriction site is working. So that is how you can get the number of total fragments upon partial digestion. The question says upon partial digestion, which means that not all the fragments were equally digested at the same point of time. So the right answer will be C. uh, could I make myself clear?
Yes, yes, thank you.
Yes.
Next slide, please. Okay, so now DNA sequencing, we know that for many research purposes, for large genomic DNA library construction, for anything, for further like even battling out COVID, we need to know the sequence of the DNA first. If we do not know the sequence of the DNA, we are not able to find counteractive measures for like designing antibodies or small targeted drug molecules against SARS-CoV-2. So anything you need to know the sequence of the DNA. So this is a very age-old method that has been coming on. So there is generally three generations of sequencing: the first, second, and the third generation. The most primitive type is the first generation, which uses the termination of the DNA sequence using dideoxynucleotides. So you have four different dideoxynucleotides for cytosine, thymine, adenine, and guanine. So the gist of this fact is that once these particular dideoxynucleotides are attached at the end of a particular read that has been set, uh, the further amplification or the further addition of nucleotides is stopped. So if you can see that in the diagram given below, the C is blue in color, the T is yellow in color. There are no further extension beyond the C or T or A or G due to the fact that they are dideoxynucleotides. So once you have gotten such many reactions where these dideoxynucleotides have been stopped, what you do is you set these number of fragments in a linear fashion, and you read the fragments in a base by base basis, starting from the smallest uh, oligo that has been stopped. So you maybe you have this CTGAGC, then you are going to read CTGACTCT. You are reading one base at a time and conferring the sequence of the DNA. This is the most primitive method. This is Sanger sequencing. Next, please. Then comes the second generation sequencing. Now, this second generation sequencing is far more sophisticated and less time consuming as compared to the first generation. What this does is this is based on a parallelized amplification. So you have your gene of interest that you want to sequence. You anneal it to a particular primer or a probe that is already set up in a chip. And once your DNA, this particular DNA, it binds to this probe, and then you are going to sequence this DNA from a top to bottom basis in the way that they have complementary ends at both of the sides. And when this single stranded DNA, it forms a bridge like structure, as you can see that in the first, second, third, fourth, fifth step, the fifth image, the this entire single stranded DNA, it has been bent into a bridge like form. So the two ends are tethered, and you start synthesizing a complementary DNA with all the given oligos from either of the ends. So once this a secondary complementary sequence starts coming up, you keep on denaturing and reannealing and making multiple such bridges. And these each single bridge, every time it forms, the number of oligos that are going, that are being accounted for, and that is how you are getting to, you're amplifying the number of the template gene that you have, and you are getting the a greater throughput result of this is how the sequence is going with each nucleotide addition. And thus you have a cluster of DNA that gives you the entire second generation DNA sequencing. This is a far robust method than what used to be in the first generation. And the next one, please. And this is the third generation sequencing. The third generation sequencing is it's a, what will I say? It's a it's it's more of a bionic process. It takes into account a protein such as the one here. This technology is called nanopore. This kind of protein, it's a motor protein. What it does is it breaks the double-stranded DNA into a single-stranded DNA, and once this single-stranded DNA is threaded into this motor protein, uh, with each base passing through the this unwinding enzyme, there's a voltage fluctuation that corresponds to either A, C, T, or G. So you get a real-time output of which particular base is passing through that unwinding enzyme, and that is how you get the readout of the DNA sequence that you're gonna have. This is the third generation sequence, and this is a very
Excuse me.
yeah.
But then the second generation sequencing, how do we come to know which nucleotide is present? Like,
It is also based on the D-deoxynucleotides only, but sometimes D-deoxynucleotides used to be old. The initial second generation used to use such D-deoxynucleotides, but now they have fluorescent nucleotides corresponding to fluorescence of each AT or C or G. So every time a single nucleotide is added, maybe A has a red fluorescence, T has a green fluorescence, G has a blue fluorescence, and C has a yellow fluorescence. So each time new your bases are added, you will get the fluorescence readout accordingly.
okay, thank you.
So yeah, this is a patented technology, millipore nanopore, and this gives the um, real-time output of the DNA sequence that is being going on. Next, please. Okay, so this is the summary of the Sanger's method or the first generation sequencing. There is denaturation of the DNA, yeah, using heat. Then you make amplification of the single stranded DNA. You make multiple copies of the segment. You attach a primer to the end, and add to 4 polymerases of the solutions. Like 4 polymerases of the solutions are, one of the solution contains dideoxy A, the same for CTG. Now you grow the complementary sequence until they terminate. So like maybe you have if once the polymerase starts acting, you'll get a certain point where this particular reaction will end. At one point, maybe it ends with a G at the third point, and then there's a G at the sixth point. So one reaction will end at the third position. The second reaction in the same tube will end at the sixth position. So now you've got places for, and you have got the locations where 2 different G's will be there. The next is like it goes on same for every other nucleotide. Now what you're gonna do, you are gonna denature these grown chains, and then you're gonna read it on the electrophoresis from a top to bottom fashion, so that the smallest fragment is, or the first read is in the lowest panel, and then it, with each increasing base, single base, you get the sequence. But this sequence that you have already gotten in the Sanger's method, this is the complementary sequence of the original gene that you had given, because you are making a complementary strand with the dideoxynucleotides, and thus the real sequence will be what is the sequence that you get complementary of the readout. Somebody has raised their hand.
Yeah, excuse me.
Yeah.
Can you just again explain what's the role of the polymerase solution in this whole process?
Okay, you are giving a template DNA, right? And from the template DNA, you are attaching a probe, and the template DNA is being copied into its complementary strand. So if you don't put up the polymerase, you cannot incorporate the dideoxynucleotide into the into the growing fragment. So the polymerase not only adds your dideoxy at the terminal end, but each nucleotide is being added in a step-by-step fashion by the polymerase. So it is not a, it is think of it as in a counterintuitive fashion. You are giving the template strand. The template strand is being copied in a single base to base fashion, and then it is terminating at a single end in a chance factor that okay, in this tube there is dideoxy G, so dideoxy G is coming and terminating the reaction at the third position. So this addition to the complementary strand is done by the polymerase. Did you understand?
Yeah, okay, thank you.
Yeah. Yeah, this is, can anybody tell me how would you read the sequence, like from where we do start, and how is the sequence read? Just anyone, which one will be the option?
Option
C.
A.
Yeah, hello.
C.
Is it option A?
No. So see, this is the readout that you are getting. Okay, now
Option
someone
D.
who was asked. No, no, not option.
It's C, I guess.
It is C. So you are going to read the sequence from bottom to top fashion, and then you are going to complement this strand, because that was the original sequence of the DNA. This DNA that has been constructed, this is just the complementary to the real sequence. So that is how you get the readout from a Sanger's method. Next, please. So this is another question. What will be the gel image of the following strand if we use only DDTP? Can
Excuse me.
anybody? Yeah, yeah, yeah. Hello.
Just can you roll on to the previous slide?
Actually, I got only 6 minutes left. You can get back to us at 9:15. I think you people
Oh,
have your test, right?
okay.
Yeah. So, anyone, any guess what will be the gel image for the following template strand? I think you'll have to work it out. You do not have so much time. Gautam, just show them. And the next slide, please. Yeah. So this is just an interesting fact that Neanderthals have contributed to 1 to 4% of the DNA in the humans of Eurasians. The term has been concluded by whole genome sequencing and looking for the specific sequences. Matches have, like, this is basically that how we use DNA sequencing to answer all the evolutionary aspects, and like uh, next genome sequencing, next generation sequencing. These methods are used to answer many of the fundamental questions of how we are evolving and everything, because such technologies are helping us to. This is an application of such a technology. Next slide, please. And so this is you have been given an endangered panda species. Now you have to clone a particular gene from that panda to preserve it. So what could be the schematic to the various steps to perform this experiment? So any ideas? Somebody just give me a small idea. What are you going to do? How are you going to clone? Anything? Start. Where are you? At least where are you going to start?
Like wherever that gene is different for the panda, and like first of all, I would find some organism that has a nearly same gene, and the different part I would just take it and amplify it and grow.
Yeah, it's fine, but you are not going to take it from a different organism, but you are going to take it from the panda itself.
Like I'm gonna take an egg cell from the panda, I'm gonna enucleate it and have the somatic cell take its nucleus and put it in that egg cell and then like culture it.
Yeah, close enough. Just so, like you are along the right lines, and just show them the image, Gautam. So here you recover the oocytes, and from there you take out the chromosome, inject into a enucleated oocyte, like you said, and then you develop the embryo. Embryo is transferred into a recipient mother, which is surrogacy, and then you natural mating or mate the cloned individual, and the second generation of the offspring and the normal phenotype can be seen. So this is how the entire genomics and the recombinant DNA technology it comes in tethering into studying evolutionary aspects or wildlife conservation or even detecting crimes and everything. So these are the total aspects. I think we have 4 minutes time, and this is all we had to give. Any doubt?
Brother, could you
Yeah,
just go into that, like the first question or gel electrophoresis where
yeah,
we detected the Sanger method used this?
yeah. This one, right?
Yeah, please explain in a little detail.
In little detail. Okay, so you are going to read this particular gel in a top to bottom fashion. Okay, so your first DNA, first nucleotide is going to be DDA, right? Can you see the smallest band is in the DDA lane?
Yes.
Then you are going to go single base up. Your next, what should be your next
G.
base? Yeah, right.
DDG.
Then the other next to that,
DDT.
right.
But
Yeah.
brother, I understand this, but shouldn't the actual DNA be the complement of this, as
Yeah.
this
Yeah,
is
yeah,
the
yeah.
result of?
That is what you will get this sequence, but the actual DNA is just the complement of what you get.
So the question was, what is the sequence of the original strand of DNA? So how is D option correct?
How is D option correct? Okay, I did not see that.
The question asked, what is the
Yeah,
sequence
yeah,
of
I get.
original strand?
Maybe there has been some mistake at the end. I'll have to work it out. But do you get the concept? Like if this is
Yeah.
everyone, I do. Please take note that if this is wrong, once you get the sequence of the DNA, the original sequence of the DNA will be complementary of what you get of this readout. Is it clear?
Excuse me, but I guess it's right because if we take from top to bottom, it's AGTCA, like the C option, and D option is its complement, but in the opposite sense. If I start from the 3 prime direction, you can see that.
Yes.
So
But
student, it's all Samit, it's
Yeah,
already 8:59. I think they should go for the quiz and they will join us back at 9:15. We'll wait for them.
yeah. It's fine. You just get the basic idea of what it is. Everyone.
So students, you can go and join the quiz here and we'll
Yes.
take you at 9:15. We'll wait for you.
excuse me, can I ask a doubt till then?
Sure.
Yes, please. Can you just roll back to the slide of that Sanger sequencing one?
This one.
Yeah. So here, as if we read from down, it's like 5 prime AGTCA GGCTA 3 prime, right? This is
Mhm.
the sequence that we get in the Sanger's method. Now we have to write the complement of it, right? for our
Yes.
original strand of DNA. So
Yes.
our options, if we see the third option is from this, like the
Mhm.
Sanger's, strand. So if I
Okay.
write the complement of the C option, so
Mhm.
like shouldn't it be like it will start from the 3' direction always? That's what I want to ask.
So see, the thing is we always read these sequences from the 5' end, and you can see in this part that from the gel part, you can see that the sequence is starting from 3', and you can see for this DNT base, you're getting first of all you're getting T, then you're getting A, and till G and A till the end, that is the 5' part. So if you
Okay.
put this sequence, so it starts from the 3' to 5'. So this is the actual sequence that we got, but we read sequences from the 5' end. That is the starting position. So that is why the complement of this will be the original part. Although this is also original, but we start from 5' end, and that's why we put the complement of this part.
Okay. And
That's why.
and in the Sanger's, this whole setup that we have, how do you know that the upper one starts from 3' and the lower one is 5'? Like it's in the method is like set up in that way.
So actually, yeah. So the method is in that way only. So when you read these sequences in the detail, so you will get those things. Or we have designed. So see in this part. So this is the gel part, and this is on the left hand side. You can see that sequence. So this we have put this intentionally actually just to get you so that we can know that how much you have got from this part actually.
Okay.
Yeah. Because whenever you see the DNA sequences, it starts from 5' to 3', and then we do this complementary and other things also.
Okay. And if like our sequence starts from 5' and to 3', it doesn't matter. It starts either with the A or G or T or C. That
No,
doesn't
no,
matter.
no, no, no. Those nucleotides can be random.
Okay, then it's
So,
fine.
so there is no hit and trial that it should start with A, T, G, and C. It can be any.
Okay.
So anybody? So how's the quiz actually?
It was nice actually. Large one. It could be solved.
So any further questions from this part? We have 10 minutes to go.
Can you share the PDF of this like whole tutorial right now so that we can see if we have any questions?
Sure, sure. Just after this session, I'll upload this tutorial in the file section in Teams. You can read from there.
Okay.
So did you get the other files also? Because after every tutorial, we used to upload those tutorials. Did
Yeah,
you
yeah,
get
we have.
them? Okay. So if you have any details, so we'll post those details in the MS Teams. So if you have also any queries that you cannot ask in the tutorial, and sometimes after this tutorial you remember that you forgot to ask that thing, you can mention in the chat box. We'll address your queries from the chat box on the MS Teams.
Excuse me.
Yes.
In this question itself, brother, like, um, the D option was correct or was it wrong?
It is correct.
It was correct, now.
Yeah.
And how are we naming it 5' and 3'? Just repeat that once again, please.
Oh, okay, okay. So see on the left hand side, just do not see the question on the answer. Just focus on the left hand side, the gel part. So we have in the gel part we have mentioned that the sequence is starting from 3' to 5', and on the 3' you are getting TA till GA till the 5' end. So this is the actual sequence that you're getting from this gel. So we read these sequences from the 5' end. So what will be the complement? 3 opposite to the 3' will be the 5'. So that's why we are getting a 5' to 3' end. It's simple.
Acha, हाँ, got it, got it.
Got it?
I just forgot that the two DNA strands are written in opposite directions, like 5 to 3 and 3 to 5.
So we are asking that thing only the original strand. So this is although this is also original, but the sequence starts from the 5' end. So that the complement will also be the original of this part.
Got it. Thank you. Yeah.
Because DNA is complementary, na? So one we have one and template one is the other part. So that's why.
Can
So
you
if
tell the
you have
difference between forward primer and reverse primer?
what?
Difference between forward primer and reverse primer, and like how do we, get to know which one is one?
So those actually you have to design those primers based on experiments. So if you have the sequence starting from a 5 prime and if you have the sequence starting from 3 prime and so those things you have to know prior hand. So we have we have the experiments from which we got we from which we got this so we get the information that which premise we need. So those things are in detail actually. So we will not covering those part in this part in this tutorial because for that you need the prior knowledge of the sequence. So you must have the somewhat knowledge of those sequences before adding those primers.
But we got a question in the quiz.
So that you have if because we have not covered in this part, so that must be in the that the textbook that we have mentioned, Campbell.
Okay.
Yeah.
Excuse me, are primers supposed to be complementary to the template DNA sequence, or are they supposed to be like exactly like the template DNA?
No. So they not be like exactly that part. So they have the nucleotide. So whatever they will get complement to them, they will synthesize the band.
So primers aren't supposed to be complementary to the template DNA. They are supposed to be like they have the same sequence, which is like actually the part of the template DNA.
Not exactly the same part actually. So if you have if you have one sequence and you are adding primers in that, so the primers so what we know, so prior hand we get the glimpse of the organism. So what is the genome of that organism, and accordingly we synthesize those primers. And it should not be it should be that exactly the same part actually. So if we have the in the primer you get ATGC. So if the corresponding to A you are not getting anything, then it will not synthesize these parts.
The primers, okay.
Yeah. So actually, when you read these things in detail, so you will get gaps also in this part. So that you will. So those are the details of that part. So which will be not covering in this like tutorials. So we are just giving the glimpse of how these things are working actually. So in today's tutorial, so you got all the questions from this DNA part also only. Anybody?
Can you repeat, please?
So I'm just asking that in today's quiz you got all the questions from the DNA only.
Yeah, DNA about it.
Okay.
It was based on lecture 4.
Yeah, lecture 4. So was it easy for you to attempt or a bit difficult?
It was okay.
Okay.
Like based on lectures, you can like attempt around easily
Yes.
7 to 8 questions, I guess.
So that's why I told you this thing earlier also that tutorials and classrooms are more than enough. And if you need the deep knowledge of how these things are actually working at molecular level, then you can follow that book actually. So book is for your reference only. So we are just picking some important topics that can be of interest to all of you. And if you need if you dig deeper in the subject, then you can refer those books.
Excuse me, brother.
Yeah.
Could you just like explain the DNA array thing? Micro
DNA.
array? DNA micro array
So
method?
that will be the topic. So we are not covering this part today actually. So that we have already covered. So we'll not discuss that thing today.
Okay.
So if you have queries related to that micro array, you can fill the query form on the Moodle actually. I think yes, they must have shared that part.
The query form is for the quiz, right?
So if you have another part, so also you can mention those things also on the Moodle.
Okay.
Yeah.
So the next lecture which we are having tomorrow, the quiz will be based on that lecture only.
So actually, yeah, right now we do not have any information about that part, and still actually we all are working on that part. So once we get any information, we'll just post it.
Check the chat. There is a message in there.
Right now,
Yeah.
let me check. I think they must Hatties
Just
must
have
have.
one day.
I think Hatties must have put them. Shashwati. Okay, then I think they have this framed that part now itself. So just refer this point, then this directly actually. I think this was message directly from the sir actually, and Shashwati just putting this message for your knowledge. So just follow this part then, because I don't have this part of this information prior. So I'm also seeing this information right now only.
Okay. By the way, what's a a decent score according to you in these quizzes?
Decent score of.
Like decent score in these quizzes that sort take, you know.
So what was the marks of today's quiz?
Out of one, I got like 0.7.
Okay, then it's fine. Actually, this 0.7 is still good actually, but it also depends upon the how many people are there attempting this quiz and what is the scenario. Because if sometimes you 0.7 is actually really nice, because sometimes the students they score very less, then we have to just normalize those things also. But 0.7 is good.
Okay, thank you.
Yeah.
Relative grading will be applied on each quiz or after the final midterms and all after the whole grading.
So this part will decide later on based on the your performance of all the students actually. That thing will sir let you know in the class maybe. So once you are done with all these quizzes and lectures, and then we'll see how to go about that part. So okay, students, it's 9:30. I'll be sharing this tutorial in file section. If you have any queries, you can write in the chat box, or you can directly write to sir also. And just do not hold any query with you. Just ask at one go so that you can go with ease and do not keep anything in mind without unanswered. Okay, guys, bye. I'm going to end this meeting.